Hello everyone, my name is Kavin Ramadoss and I'm a rising senior at Sunset High School in Portland, Oregon. And my project for the student speaker series is called Efficient Malaria Drug Discovery Methods and its Impact on Global Health. So let's just get a quick introduction about me. My name is Kavin, and I'm a rising senior at Sunset High School in Portland, Oregon. I'm also my school's science fair club president. I've also participated in varsity tennis for the last 2 years, and I'm also interested in a career in medicine in the future, specifically a physician. Okay, so now that we know a little bit about myself, let's actually dive into some of the background knowledge required to understand my project. So, malaria is the third deadliest disease in the world and is caused by parasites of the genus Plasmodium. It has over 249 million cases in 2024 alone and has a death rate of over 17%. And while this might not seem like much, the CDC confirms that malaria has developed resistance to all the four major drugs currently targeting it, making malaria drugs a high demand. And current drug discovery methods are very inefficient as they have two main issues. The first is that it takes around 10 years to synthesize one drug through various clinical trials, and it takes around $3 billion to test that same drug through those same clinical trials. So it's very costly and very timely as well. To combat this issue, I wanted to create a computational model to scan large chemical databases with over 2.4 million bioactive drugs and find five potential drug targets for any disease, not just malaria. To do this, I utilize four main steps as we can see here. In the first step, it is called front-end data collection, where enters input via a Windows form app, and then the user is given a choice between the default chemical database called Campbell, or the user can bring one as well. The second step is called molecular docking, where I take a protein structure and add a drug into this protein structure and try to see how well a compound binds to this protein structure. This score is also called the binding affinity score. And then what I do is that I rank compounds on their binding affinity score and take the best compounds. The third step is called drug-like filtrations, where I see if the compounds can actually become viable for human use. The fourth and final step is called molecular dynamics. Okay, so now let's dive into each step a little deeper. The very first step is called protein and database preparation. As for any good computational model, your data has to be clean so the computational model can understand it. So I had to do that for something in like this. So to prepare the protein, I added hydrogen atoms, removed water molecules, and added Coleman charges, and that prepared the protein. And I used the exact same methodology to prepare the database, which is the database of drugs as well. So moving on to step two. Step two is a step called molecular docking, where I take a protein structure and add a drug to it. And in this step, I decide I can determine how well a drug binds to a certain site in the protein structure, and this is denoted by a binding affinity score. Through this step, I was able to take 2.2 million potential bioactive drugs from the initial dataset and reduce it down to 32 potential bioactive drugs that had high binding affinity scores. Through this, I'm able to determine that these drugs have high binding affinity scores, meaning that they could potentially inhibit malaria. Now let's talk about the next two steps. Now that we have 3,200 drugs that could potentially inhibit malaria, we actually need to see if they can become viable for human use. To do this, I use a step called ADME, called absorption, distribution, metabolism, and excretion. And these are a set of properties to determine whether a drug can actually become viable for human use. Through these steps, I was able to take the 3,200 drugs and evaluate them for their drug-like properties, also called pharmacokinetic properties, and was able to select 10 final drugs. The last and final step is called molecular dynamics, where I'm able to understand how the protein and ligand interact in a water box molecule at a deep molecular level. Through this, I was able to select the five final selected anti-malarial compounds. Okay, so now let's talk about some of the results from my project. The five final selected compounds had exhibit favorable ADME properties, which means that they could potentially be viable for human use. Furthermore, these compounds had low toxicity and high synthetic accessibility, which means that they can be easily synthesized by a scientist in a lab. Furthermore, the molecular docking results show that the binding affinity scores range between negative 8.28 and negative 10.83 kilocalories per mole, which means, and these are all relatively high, which means that the compounds exhibit a high binding affinity score. So let's continue on with the results. In fact, let's talk about the molecular dynamics results. Post molecular dynamics, I calculated two metrics called RMSD and RMSF. RMSD is a metric that shows how much each compound deviates from its initial conformation over time, and usually an RMSD below 1.5 is indicative of ligand stability and interaction strength with the protein. Since the highest value is zero point 0.7, each of the five final selected compounds, L1 through L5, has good stability and interaction strength. Now, moving over to RMSF, the RMSF measures the fluctuation of individual atoms or residues over time to measure protein stability to see how well a compound binds. Usually, an RMSF below 1.5 indicates that the protein is very stable, and since the highest value is zero point 0.8, the protein is stable, allowing for better binding with the five final selected compounds. So now let's talk about my conclusion. In my conclusion, I wanted to talk about a few main things such as the summary, why it matters, the application, and future work. So for the summary, I developed a low-cost and integrated biocomputational framework that identifies five hit compounds for malaria. Furthermore, I started in vitro testing for all five of these compounds. And why does it matter? The framework is the first of its kind as no other commercial tool exists today. Also, the framework reduces the current cost and time of drug discovery by 50 and 75% respectively. Lastly, I proposed a new malaria inhibitor which can bind to a novel allosteric binding site. So now let's talk about the applications. The novel malaria inhibitor that I discovered can help millions of individuals suffering from malaria, and this is why it connects back to global health. Furthermore, the framework is universal and usable for all diseases, not just malaria, such as HIV, tuberculosis, cancer, and more, and can greatly impact drug discovery, allowing for many more drugs to be produced. These are some of my references. And once again, my name is Kavin Ramadoss, and you can see my email and my LinkedIn listed. Thank you so much.